creatine and protein synthesis. This is where a lot of content gets it backwards. The common claim is that creatine directly stimulates muscle protein synthesis. That framing is misleading, and understanding why it's misleading actually clarifies how creatine's muscle-building effect works. Muscle protein synthesis, the process of assembling new muscle proteins from amino acids, requires two things at a fundamental level: adequate amino acid supply and an anabolic signal telling the cell to build. Creatine doesn't provide amino acids, and creatine doesn't directly activate the primary anabolic signaling pathways the way dietary protein or an acute testosterone spike does. So where does the confusion come from? There are two documented ways creatine influences protein synthesis, and neither is the simple creatine builds muscle story that gets repeated. The first is indirect but most important. Creatine allows you to do more work, more reps per set, more sets per session, higher power output across the entire training session. That increased training volume is a more powerful anabolic stimulus than almost any supplement-driven mechanism. The downstream effect of training harder and longer, consistently over months, is more muscle protein synthesis, more satellite cell activity, more repair-driven growth. Creatine earns its muscle-building reputation primarily by expanding training capacity, and the muscle that results is from the training itself, not from the creatine directly. The second mechanism is more direct but context-specific: cell volumization. When creatine loads into your muscle cells, it draws water in via osmotic pressure, causing the cells to physically swell. This swelling creates mechanical tension on the cell membrane and changes the osmotic environment inside the cell. And those changes act as a biological signal. Specifically, cell swelling activates the mechanistic target of rapamycin, mTOR, which is the master regulatory protein for protein synthesis in muscle cells. Studies in isolated muscle cell preparations show that osmotic swelling alone without exercise can activate mTOR and increase protein synthesis markers. This is one reason creatine contributes to some muscle protein synthesis stimulation, even in the absence of training. Not enough to be a standalone muscle builder, but enough to contribute meaningfully when combined with training. Cell swelling also stimulates satellite cell proliferation. Satellite cells are the muscle stem cells that sit dormant between muscle fibers, waiting for activation signals. When activated, they divide and fuse with existing muscle fibers, adding new nuclei and increasing the fiber's protein synthesis capacity, or remain as a reserve for future repair. The osmotic signal from creatine-induced cell swelling is one of the stimuli that activates satellite cells. This is part of why creatine supplementation during a training program consistently produces slightly more muscle mass than training alone, even when caloric and protein intake are matched between groups. A third consideration that's less discussed: creatine reduces markers of muscle protein breakdown after intense exercise. Some studies show lower post-exercise levels of muscle damage markers, including creatine kinase release and myofibrillar protein degradation indicators, in supplemented versus unsupplemented individuals. The net balance of protein synthesis minus protein breakdown determines whether muscle tissue grows, maintains, or shrinks. If creatine simultaneously increases the anabolic side through cell volumization and reduces the catabolic side through protective effects during exercise, the net result is a more favorable muscle growth environment, even at the same absolute training volume. The practical takeaway: creatine is not a substitute for dietary protein. You still need adequate protein, consistently, across the day, to provide the amino acid building blocks for the muscle protein synthesis that creatine enables. Creatine without adequate protein intake is like building a larger construction crew without ordering more materials. The crew is more capable, but they're limited by what's available. Combine sufficient dietary protein with creatine supplementation and consistent training, and you've addressed all three legs of the muscle growth triangle: the signal, the substrate, and the energy capacity to generate the training stimulus. That combination is why creatine remains the most consistently effective legal supplement for muscle development in the evidence base. Not because it builds muscle directly, but because it reliably improves every input that does. Creatine without protein is a capable engine with no fuel. Protein without creatine is abundant fuel with no engine at full capacity. The combination is the whole machine.